I think it is both important and interesting to see how the ultrastructure of the skeletal muscle fibre is specialised for contraction, apart from knowing about the structure of the myofibrils and myofilaments. This model represents the skeletal muscle cell, or more commonly referred to as a muscle fibre. It's orientated as if the muscle fibre extends from left to right. It makes far more sense to call it a muscle fibre than a muscle cell because it's such a long structure. Similarly, we often call a nerve cell a nerve fibre. Here you can see the nerve fibre passing through the endomysium, and it will also then pierce the external lamina to make contact with the muscle cell membrane. This contact is a very specialised structure called the neuromuscular junction. In a skeletal muscle fibre, the cytoplasm is referred to as the sarcoplasm, and the plasma or cell membrane is referred to as the sarcolemma. Myofibrils dominate the sarcoplasm. They're these long cylindrical bundles of myofilaments about 1 to 2 micrometers in diameter. You saw them earlier in transverse sections through muscle fibres. Mitochondria and smooth endoplasmic reticulum lie between the myofibrils. This structure here represents one of the many nuclei associated with this muscle fibre, lying underneath the sarcolemma. But remember also, a few nuclei of the satellite cells, which are not shown here, would lie just under the external lamina. A nerve impulse, or rather an action potential, travelling down this branch of a motor nerve reaches the neuromuscular junction and depolarizes the sarcolemma to initiate a wave of depolarization along the muscle fibre. All along the muscle fibre, the sarcolemma invaginates into the sarcoplasm, forming this system of transverse or T-tubules. They circle each myofibril almost in the region where the A band and the I band meet in each sarcomere. So, the wave of depolarization instantly passes to all parts of the length of the myofibrils and all the myofibrils, even those most deepest in the muscle fibre. The agranular or smooth endoplasmic reticulum is called the sarcoplasmic reticulum and is specialised for calcium storage and release during muscle contraction. Adjacent to the sides of each T-tubule is an extensive series of channels of the sarcoplasmic reticulum called terminal cisterns. This arrangement constitutes a triad. Now, this intimate contact of the sarcolemma with the sarcoplasmic reticulum causes it to depolarize, and calcium ions, which have been concentrated within the cisterns, are released from the sarcoplasmic reticulum to cause uniform contraction of the myofibrils. When depolarization of the membrane has ended, calcium ions are then pumped back into the cisterns of the sarcoplasmic reticulum and muscle contraction ceases. The mechanisms of muscle contraction is beyond the curriculum of this histology course, but no doubt you'll learn about this fascinating biological process in your physiology course.